So our polypeptide chain has just come from the organelle ribosome. And once it hits its termination sequence, as we remember, termination, this polypeptide chain has to travel to the rough ER, the organelle rough ER. And this step is called post-translational modification. Post-translational, just coming from translation, modification, having to be folded and formed into its 3D shape. So the folding and forming of its 3D shape happens in the rough ER. Once that's done, it has to travel via a vesicle, because if it didn't, the folded polypeptide chain would become unfolded in the cytosol or cytoplasm. So once it's released, then it goes to the Golgi apparatus. And the Golgi apparatus is the organelle that addresses it and packages it to where it needs to go. So once it's done in the Golgi apparatus, it has to go through a vesicle again, carrying the functioning protein out of the cell. And once it goes out of the cell, that's called exocytosis, exit. And it exits the phospholipid bilayer of the cell into the extracellular fluid outside the cell. So cytoplasm in, ECF outside the cell. And in our example that we had, the pancreas produces insulin proteins to stabilize blood sugar to maintain homeostasis. But to make those proteins, it has to go through all 4 of these steps and exit the cell.